Going to be talking to you about taxonomic and functional analyses of intact microbial communities thriving in extreme astrobiology relevant anoxic sites, courtesy of Bashir et al. 2021. While Bashir et al.'s research goals were multifaceted and it's difficult to distill their work into a single research question, their paper can be unified by the guiding inquiry of using extreme terrestrial environments as analogs for extraterrestrial settings and determining what analysis of such microbial communities reveals. One way to understand life is by its limits, and by selecting several anoxic environments in the most extreme conditions on Earth for microbial analysis, these terrestrial analogs of extraterrestrial environments allow inferences to be made of where life may exist outside of our planet. The work of Bashir and her colleagues is notable for being the first comprehensive analysis of oxygen-free terrestrial environments and also being the first study to use propidium monoazide, also known as PMA, to mask background DNA from dead microorganisms. This allowed a novel analysis of the intact and living microbial community and the isolation of 31 model microorganisms to be used in future astrobiological research. Beyond elucidating these highly valuable model organisms, the study also deciphered molecular principles of resistances, genomic and metabolomic characteristics of extremophiles, and will contribute to the optimization of automated life detection in the future. Perhaps one of the most defining philosophical quandaries unique to the human condition is pondering our place in the universe and the possibility of life beyond Earth. Propelled by sudden public and academic interest in extraterrestrial life, the 1950s marked the formalized inception of astrobiology. In the first efforts of astrobiological space exploration in the 70 years that followed, the scientific discipline is still faced with defining two seemingly simple parameters. One, the conditions of a planet that would make it conducive to life or habitable, and two, biological signatures or biomarkers that can be identified on a proposed habitable planet and indicate the presence of life. Defining both of these parameters is complicated by the fact that there is no standard of comparison for life outside of the Earth. The physiochemical properties of biomolecules result in most living organisms only being able to function with a certain range of conditions, and this range of conditions considered conducive to life was long understood as being relatively restrictive. This perpetuated an understanding of life as fragile, requiring an abundance of liquid water, mild temperatures, protection against radiation, and a multitude of other highly specific conditions to survive. The fragility of life has been greatly challenged by the discovery of extremophiles, though, organisms persisting in environments previously thought to be inhospitable to life, ranging from hypersaline lakes and hyperacidic hot springs to sub-zero polar environments and nuclear reactors. Extremophiles have widened the understanding of where life can survive on Earth, and by extension, given insight into where life could be elsewhere in the universe. Beyond expanding the number and variety of extraterrestrial locations that could be targeted for future exploration, the study of extremophiles has refined the processing of environmental samples for the detection of viable life forms, identified new biomarkers, and enhanced understanding of cellular evolution, all of which have played critical roles in the continued pursuit of astrobiology. Initiated in 2014, the Mars Analogs for Space Exploration, or MASE project, is a collaborative research endeavor that seeks to understand how life can adapt to extreme conditions and assess the habitability of Mars. Working in service of the MACE project, this study, undertaken by Bashir and her colleagues, filled in critical gaps of knowledge that elucidate whether the combined environmental stresses experienced on Mars are compatible with life and whether a record of that life could ever be detected. While a large number of analog environments have already been investigated as terrestrial analogs, they are mostly oxygenated and thus are not representative of extraterrestrial environments that are either oxygen-free or contain extremely low abundances of oxygen. Motivated by this gap in the literature, Bashir et al. Set out to answer the following questions. Which bacteria and archaea are alive under extreme anoxic environmental conditions? Which functions do they possess? Can we cultivate them? Is there a core microbiome of all sites? And lastly, are there general principles of anaerobic microbial communities with impact on the habitability assessment and the search for life on Mars and beyond? By nature of the study's use of a broad combination of anaerobic cultivation and molecular methodology, the researchers did not have a hypothesis. Since no single terrestrial analog site contains a combination of all extraterrestrial physical and chemical conditions simultaneously, a representative set of terrestrial environments were analyzed. Samples were retrieved from 13 selected sites at 5 sampling locations and chosen based on their astrobiology-relevant chemical and physical characteristics, as shown in this table. For selection, an emphasis was placed on sites with low water activity, low temperature, and acidic conditions. All selected sites have the parameters of oxygen limitation or anoxic conditions and restricted availability of complex and organic nutrients. One glacier environment was added for comparison, for which the samples were retrieved during a Mars landing simulation of the Austrian Space Forum. In summary, the sampling sites included two sulfidic springs, one subsurface hypersaline environment, one acidic lake, one acidic river, two permafrost environments, and these are all collectively later referred to as the mace environments, and the glacier environment for comparison. After selecting the sites, sediment, water, and soil samples were obtained during sampling campaigns performed in 2014 and 2015. Prior to DNA extraction, cells were treated with propidium monoazide to discriminate between viable and dead cells, with the exception of the hypersaline environments, as PMA treatment is inefficient at high salt concentrations. Library preparation and sequencing was then performed, beginning with two distinct PCR reactions with primers carrying an alumina MySeq compatible barcode adapter. The first PCR targeted bacterial and archaeal 16S rRNA genes, and the second targeted archaea only. Each sample was indexed with a unique barcode sequence and then subsequently sequenced using the aluminum MySeq device. Reads from these amplicons were then processed using R, and sequences were assigned to a taxonomy using the RDP training set classifier version 14 and the Silva version 123 classifier. Shotgun metagenomic analyses were performed on 6 selected samples from the sulfidic springs, acidic lake, and permafrost sites, respectively. For gene-centric analyses, MG-ROST and MEGAN were used, and the resulting taxa and functional gene tables were visualized using Calypso. The genome-centric analysis was supported and curated by a gene-centric approach, including further analysis in MEGAN. Bashir et al. First focused their data analysis on the archaeal community. This graph captures the archaeal profile of intact microbial communities from mace environments, specifically the taxonomic profile obtained using the archaea targeting primer. The left bar chart shows the relative abundances by family level, and the right bar chart by phylum. As indicated by their absence from these graphs, no archaeal signatures were obtained from the glacier samples. 787,842 archaeal reads were obtained using the archaea targeting the archaea targeting primer set, resulting in 1,502 archaeal ribosomal sequence variants, furthermore referred to as RSVs, and high variance across samples. The lowest number of sequences was retrieved from sample acidic lake SS3 with 72 reads, and the highest read count was obtained for sulfidic spring IM with 106,622 reads. Interestingly, no archaeal RSVs were observed from the samples obtained from the glacier, which indicates that the level of archaeal 16S rRNA genes were below the detection limit or that the primer match was insufficient. The majority of RSV counts were assigned to the phylo Uri archaeota at 39.3%, Thalma archaeota at 22.7%, and unassigned archaea at 20.8%. Signatures of Uri archaeota were detected through all the archaea positive samples except for the acidic lake SS1 sample. Notably, 99.9% of the sequences detected in the acidic lake SS1 remained unclassified within the archaeal domain which may represent a new taxon. Using a universal primer set to target the entire microbial community, the researchers amplified both the PMA-treated and untreated 16S rRNA gene pools, eventually obtaining 1,523,276 sequences and 15,945 different RSVs post-processing. The intact microbial communities were generally characterized by proteobacteria, which had a mean total amount of RSV counts of 27%, followed by actinobacteria at 11%. Directing your attention to the microbiome diversity visualization on the left, indicated by the letter A, the samples from sulfidic springs showed the highest diversity when observing all microbial signatures, whereas the lowest diversity was observed in samples from the hypersaline environment. Similarly, the highest archaeal richness and diversity occurred in the sulfidic spring biotopes, whereas samples from the acidic lake contained an overall very low archaeal richness and diversity. PMA-treated and untreated samples did not differ significantly in archaeal richness and diversity. In the lower figures B and E, the beta diversities of the archaeome and the general microbiome were visualized with principal coordinate analysis plots. Despite being obtained from geographically remote sampling sites in Iceland and Spain, the microbial communities derived from the acidic environments clustered in both plots, along with the sample from the hypersaline environment. Additionally, permafrost samples, sulfidic spring samples, and glacier samples grouped separately into their own clusters. Overall, the highest taxonomic diversity was detected in sulfidic spring samples and least in the permafrost samples. The signatures of a broad diversity of facultatively and obligately anaerobic and extremotolerant extremophilic microorganisms were elucidated in the datasets. These included methanogens, halophiles, acidophiles, and multiple taxa associated with psychrotolerance or growth in low temperatures. Relevant anaerobic metabolic capabilities were also identified, including iron reduction, sulfate reduction, nitrate-dependent iron oxidation, and methanogenesis. Additionally, signatures of spore formers were found in all environments. The researchers were also interested in whether the sites shared certain microbial signatures. The intact core microbiome of all sampling sites comprised 4 taxa. 2 of them could be classified on the genus level, specifically Polybacter, with 12 different RSVs present in all sampling sites. The core microbiome was predominated by unclassified microbial signatures, Streptomyces, Mycobacterium, unclassified PVC group organisms, unclassified Proteobacteria, and Bradyrhizobium. 3 samples were also selected for metagenomic sequencing, with each sample having 2 parallel fractions processed, one PMA treated, the other untreated. The taxonomic information derived from the metagenomics dataset was congruent with the findings from the amplicon sequencing approach. However, the permafrost sample showed a higher contribution of acidobacteria and the archaeal tax superphylum when PMA treatment of the sample was performed. The signatures of alveolata and amoebaza were also found to be reduced after PMA treatment in the acidic lake samples, indicating their possibly high contribution of free DNA. Comparative analysis of all samples revealed the I'm so sorry. Comparative analysis of all samples revealed the diversity of microbial functions to be significantly higher in the permafrost sample and lowest in the sulfidic spring sample. The most abundant enzymes/functions detected in the shotgun metagenome dataset were integrase, glycosyltransferase, cation efflux system protein cus-A, long-chain fatty acid CoA ligase, and decarboxylase. Most excitingly, the functional profiles of the microbial communities clearly mirrored the extreme physical and chemical characteristics of the sites, such as the permafrost sample being characterized by an increased turnover of amino acids, increased cold shock response, dormancy, sporulation, iron acquisition and metabolism, stress response, and sulfur metabolism, and the sulfur spring sample being characterized by quorum sensing and biofilm formation, respiration, cofactor formation, and nitrogen and phosphorus metabolism. While the sites used as analogs in the study were indeed extreme environments for the microorganisms, as indicated by the core genome of the 3 model isolates involving stress-revealed genes, all levels of analysis still indicated a strong dependency on the terrestrial microbial communities on complex organic matter. Being the first comprehensive analysis of oxygen-free terrestrial environments and also being the first to use PMA to mask background DNA, this study provides significant contributions to optimizing research for future Mars analog sites by adding resolution to the definition of habitable extraterrestrial environments and providing important information for future life detection missions. Bashir and her colleagues elucidated 2 general functional principles of all analog microbial communities: resilience against ionic strength and freezing, both of which are potentially representative of extraterrestrial stressors. While the study's primary strength was in its ability to eliminate terrestrial oxygen contamination in its chosen sites to match extraterrestrial conditions, one significant limitation was the omnipresence of organic compounds. Another limitation was the insufficiently low DNA concentrations that prevented many samples from being included in the metagenomic approach. While this study did contribute novel insights into the microbiology of analog sites, several research goals remain unaddressed. Most significantly, a need for the evaluation of the impact of terrestrial organic molecules on the biology of analog environments for astrobiological research. In spite of its limitations, the study has exciting implications for the tenacity of life and its existence in places previously thought uninhabitable. All right, so to get our discussion started, firstly, what effect could organic compounds have on the profile of microbial communities? This is something the authors were really, really concerned about, but they never really explain why. So I'm curious what you think. 2, is there risk to the study of terrestrial analogs, creating a geocentric definition of life? And by this, what I mean is that if we're only understanding life by the way that it exists on Earth, then is there possibly a risk of not being able to recognize life elsewhere in the universe? Because what if it doesn't resemble anything that exists on Earth? Or do you think it's still valuable to have this kind of research in conjunction with exosolar exploration? And three, what are other terrestrial environments not included in the study that would be useful to study as analogs? So get creative. Four, is the search for microbial life on other planets something that is societally important? And I think this is a really interesting discussion to have because, with all the problems we already have on Earth, health-wise, biologically, socioeconomically, is it like I feel like there could be a strong argument to be made as to why this kind of research either really is still important or is not important. Especially compared to like other microbiome related research in humans. But I think it's really important, obviously. Hence me choosing this article. And then lastly, five, what could be possible applications of extremophile research beyond astrobiology? So that kind of ties into my previous question. Maybe by thinking of other on Earth applications of extremophile research, you could create a stronger argument for why it's still important. I mean, yeah, but I think the pursuit of knowledge in any capacity is always important. And yeah, here are my references. Thank you so much for listening.